Hey, it's Horner, and we need to go through the parts of DNA. And so this, as long as you kind of just follow along, you'll see where everything is. And let's start with just the whole thing. So the whole thing that you see here, so the little dots, the pentagons here, the little circles that are ovals that might have a piece cut out. You'll see kind of how things fit together. And then you get the same thing on the other side, but notice that this one points up and this one points down. So what is all this stuff? The thing that you see that's in this box, which is also over here, is called a nucleotide. It's got three parts. First part is a phosphate. And you'll notice it's just a phosphate group. And this is just super simplified so that you can see it. Phosphate group. It also has a sugar. And this sugar is ribose. Or you could look at it and say, oh, look, it's a 5-carbon sugar. And attached to that is a base pair. And so we'll see the nitrogenous bases. And all that means is they have nitrogen in them. So adenine, guanine, thymine, and cytosine. So those are the 4 DNA nitrogenous bases. And you'll notice that when we put a bunch of nucleotides together, so we just stack them, so you'll notice you have this one, and that's the same thing here. And if I stack them using these bonds called covalent bonds, those are the solid bonds, so they're everywhere on here. Then we make the sugar phosphate, because there's the sugar and there's the phosphate, and it kind of looks like a backbone. How all of the different vertebrae are in your body, we also have all these sugars that kind of line up too, and they're connected by these phosphates. So let's take a look at the next thing. This one is purines and pyrimidines. And the best way to kind of think about this is purines are pure, so there's your purine, as, and notice A is capitalized and gold is capitalized, because A stands for adenine and G stands for guanine. Now, how do we know that these structures are purines? Well, remember, it's better to have 2 gold rings than it is just to have 1. And so purines have 2 gold rings. Pyrimidines, on the other hand, they look like a pie or they look like a cake because they're basically just 1 ring. And you'll notice that cytosine and thymine are the 2 that we think of when we think of DNA. But then uracil, that is used whenever we look at RNA. And we're not worried about that yet. We just want to worry about cytosine and thymine. So 1 ring is basically just 1 pie or 1 cake. And that makes it a little bit easier to see. Now, how do they pair up? That's the other thing that we really need to take a look at. So we're going to move this back down and we're going to take a look at that original picture again. Notice that A and T always are together. And it can either be A on the left, T on the right, or T on the left and A on the right. A bonds with T. And that's a super simple word. If you think about it, it's just at. And because it's a simple word, notice that it only has a double hydrogen bond. So there are 2 hydrogen bonds with this. When we look at the next to the bond, cytosine bonds with guanine. And the way that I like to try to remember it is I just take C and then I flip G backwards. And if you push them together, notice it looks like that triple bond that occurs between C and G. And remember, it doesn't have to be C on the left always and G on the right. It could also be G on the left and C on the right. Hopefully this little video helps you a little bit with those things, and that's it.